They are some of the ocean's smallest inhabitants. And on coral reefs, microorganisms are copious creatures.
Microbes are very prolific on the surface of corals, both in healthy and diseased corals.
But in a world that's invisible to the naked eye, drastic changes are taking place.
Whether it's Australia or the Bahamas or the Florida Keys, all the scientists see this microbial power shift. With healthy corals, when temperatures increase, pathogenic bacteria go up and the beneficial bacteria go down.
Just like when we get stressed out, we're more likely to get a cold. When you stress out these corals, they could potentially be more susceptible to bleaching or getting a disease.
Throughout Florida, scientists painstakingly work to identify key players within this microbial community.
It's estimated that you can only culture 1% or less of the bacteria that are out there, but yet corals have thousands of different kinds of bacteria associated with them.
While some of the bacteria found on corals are important to their health, others can lead to disease outbreak.
This bacterium was killing the coral. We could hypothesize, based on what we knew about the bacterium, that it might be coming from humans. Usually we hear about pathogens from wildlife to humans. Examples are HIV, swine flu, avian flu. But we haven't had the reverse, and so this is a new disease mechanism that we're not familiar with, especially in the marine environment.
With experimental sites in the lab and in the sea, what will microbes reveal about coral health? How do human activities impact the microbial balance on the reef?
Major funding for this program was provided by the Batchelor Foundation, encouraging people to preserve and protect America's underwater resources, and by Divers Direct, inspiring the pursuit of tropical adventure scuba diving.
They help protect our coastal communities from massive storm surge. Their beauty and bounty fuels a multi-billion-dollar tourism and fishing economy. Coral reefs are the underwater oases of the oceans. But rising water temperatures and pollution are taking their toll on the health of these gardens of the sea. In Summerlin Key, Florida, researchers from Mote Marine Laboratory are studying the microbial communities that surround corals.
We're leaving to go offshore about 5 nautical miles to the famous Lou Key Special Protected Area in the Florida Keys National Marine Sanctuary.
Leading today's excursion is Dr. Kim Ritchie, manager of the Marine Microbiology program at Mote Marine Laboratory.
I'm most interested in corals that are reef-building corals, some of the bigger corals and some of the corals that grow fast and actually build the reef structure, because these are the ones that are more susceptible to diseases.
Dr. Ritchie has been studying coral microbes since the early 90s, a period of time that coincided with visible changes on reefs throughout the Keys.
That happened to be at a time of increased ocean temperatures and coral bleaching events and increased diseases.
Water temperatures rise, they get higher than usual. Corals bleach, and by bleaching, I mean they lose the coloration, the plant type of algae.
These symbiotic algae live within the coral's tissue, but if the algae are absent for too long, the corals can die. For the past 7 years, Dr. Ritchie and her staff have traveled to Looe Key to collect mucus from various corals.
And we will agitate very gently the coral surface, and it'll give off this mucus. The surface slime, this protective layer that harbors a lot of the bacteria.
Experts are hoping that a closer look at microbes living in the mucus will provide better clues about the health of these marine invertebrates.
Corals are very ancient animals, so they have an ancient immune system. What ancient organisms often use to help them with their immunity are bacteria that they utilize to help them fight off diseases.
Over time, experts have observed a pattern of change related to bleaching events.
One thing that we know from studies that we've done since the 90s are that corals have a particular balance of bacteria associated with them. What we see is when temperatures increase, even before the corals bleach, these pathogenic bacteria populations go up and the beneficial bacteria go down.
In recent years, Dr. Ritchie has noticed an imbalance among a group of pathogens or disease-causing bacteria.
One thing that we see when corals are stressed are Vibrio-type bacteria.
Vibrio are a large group of bacteria typically found in marine environments. The most common strains are known to cause foodborne illness through consumption of undercooked seafood.
If you find the majority are Vibrios, when you culture them, that's a sign that the corals are sick. Because I was measuring monthly, we're actually able to show that Vibrio levels increase when temperatures are increasing, even before the corals show signs of distress, even before you see that they're bleaching.
Most people realize how hard it is to keep a reef tank in their home. They can't have temperatures going all over the place. They have to have good water quality. Well, it's the same with the corals here.
Scientists at Mote hope their research will continue to provide clues about bleaching events and other microbial activities. Meanwhile, other experts are studying how a unique disease is destroying an iconic species of coral. Its majestic branches resemble the shape of elk or moose antlers. And years ago, it was one of the most common corals in the Florida Keys, Bahamas, and the Caribbean. But in 2006, elkhorn corals were added to the endangered species list. Now, a devastating disease threatens what few living stands remain.
Elkhorn coral has declined by 90% since the mid-1990s. Whitepox has been a major contributor to the decline.
At Rollins College in Winter Park, Florida, coral disease microbiologist Dr. Catherine Sutherland has studied whitepox disease since its first appearance in 1996 on reefs off Key West.
It was actually discovered within a photo monitoring station that I had been monitoring for a year at that point. And I had been expecting to see increase in coral size because elkhorn coral grows relatively fast. But instead I saw a slowing of growth and then the appearance of whitepox disease within these monitoring stations. So whitepox disease exclusively affects elkhorn coral. So it appears as patches or spots of white on the coral. And the white is the dead skeleton that is exposed when the tissue dies and sloughs away.
In 2002, Dr. Sutherland and other scientists discovered the pathogen responsible for whitepox, a bacterium called Serratia marcescens.
It's a common inhabitant of human guts, so it's found in human intestines and it's found in our waste.
Strains of Serratia marcescens manifest as pink biofilm on areas such as tile grout, toilet bowl water lines, and as slime on contact lens cases.
It also is associated with respiratory tract infections, urinary tract infections, and wound infections, all common in hospital settings.
Once the coral pathogen was identified, a search began to locate the source of this bacterium.
So we started a widespread search for the pathogen in the Florida Keys, collecting samples from multiple potential sources. Human sewage from the Key West wastewater treatment facility, as well as waste of other animals like seabirds and key deer. But our genetic analyses showed that only the strain of the bacterium that was found in human sewage matched the same strain that we found in diseased corals on the reef.
Once scientists inoculated healthy corals in the lab with the matching strain, they were able to replicate disease symptoms.
We saw disease signs in 5 days. And so this was our definitive evidence that humans are a source of this devastating disease of elkhorn coral and causing whitepox. This is new because usually we hear about passage of pathogens from wildlife to humans, right? Examples are HIV, swine flu, avian flu, but we haven't had the reverse, going from humans to a marine invertebrate, in this case elkhorn coral. And so this is a new disease mechanism that we're not familiar with, especially in the marine environment.
Researchers were able to trace the human pathogen back to its source, leaking septic tanks.
Septic tanks were not designed for areas in the Florida Keys. They were designed for areas where you have soil and also where you have low population density. And the soil is extremely important because if you have a leaky septic tank, the soil acts as a filter. But in the Florida Keys, what we have is lots of people, and we have not soil, but limestone bedrock. And limestone bedrock is very porous. It's like Swiss cheese. And the leaking septic waste can very quickly move through the porous bedrock into the nearshore environment. The good news is that there is a solution, and the solution is using wastewater treatment facilities that actually process the waste and remove the microbes.
In 2001, the city of Key West switched to an advanced wastewater treatment facility. Since then, the remainder of the Florida Keys have started the process of upgrading their sewage treatment systems. Today, experts have described approximately 20 coral diseases, but only the causes for 5 of these have been identified. To get a better understanding of coral disease behavior, scientists from the University of Florida are looking at a sea anemone for help.
Aptasia is a little weedy polyp. It's a distant relative of corals. It's a good laboratory model because it multiplies really fast. It's sturdy. It's easy to work with. Unlike corals, which are protected and are really difficult to harvest in the wild, we can set up a lot of really interesting experiments with Aptasia in the laboratory.
You can simply take the coral pathogen that you've cultured in the lab and infect the animal with a predetermined dose of that pathogen. And then over time, you can monitor disease progression, which takes about a week. At the end of the experiment, macerate the polyp, break it up, and we can plate it on this and determine whether or not the cause of the sickness was actually from the pathogen that you infected your sea anemone with.
Experts hope that by testing a variety of coral microbes on sea anemones, they can better understand which organisms play a role in disease infection. Scientists also conduct DNA experiments with genes from bacteria as a potential way to biologically protect corals from disease.
In order to understand how pathogens establish on the surface of a polyp or on the surface of a coral, the first thing we wanted to understand is what are the early events in the interactions. So the first thing that a pathogen comes in contact with is the mucus.
To test how effective the mucus is in protecting a coral from harmful bacteria, researchers focus on the genes from pathogens. They try to identify the function of specific genes and how those genes interact with the mucus. Scientists hope to understand the genetic mechanisms that make pathogens stronger or weaker and how those can be manipulated in favor of the coral's health.
And the long-term goal of this experiment is to figure out a way to block or inhibit the virulence potential.
To do this, scientists isolate and genetically modify specific genes. They're testing how important the newly mutated gene is for virulence, or the bacteria's ability to cause disease. These mutations are compared to the genes of the original bacterium, or wild type.
What I do is test that mutation against the wild type in terms of its ability to growing them together. So once we incubate the plates overnight, the resulting growth looks something like this. And just using sterile toothpicks, we can pick an individual colony from the plate and then simply patch it onto this media containing the antibiotic kanamycin.
The antibiotic kanamycin is commonly used in molecular biology to treat a variety of bacterial infections. In this case, kanamycin represents the native or beneficial bacteria found in the coral's mucus, which fight off disease.
So after we incubate these patch plates overnight, then we can look at them. So the ones growing on this plate are our mutant strains.
Normally, the antibiotic kanamycin would have prevented the bacteria from growing. Instead, the mutated gene allowed the bacterium to colonize the plate.
So this mutant is now going to be resistant to the antibiotic kanamycin, and those that didn't grow are a wild type. This will tell us how competitively fit the mutant is compared to the wild type.
In this case, the mutation caused the bacteria to potentially be more harmful to corals it might come in contact with. In the future, more tests will be performed to identify which mutations can do the reverse and block disease growth.
Once we know which behaviors the pathogen relies on for the infection of the coral and the establishment on the mucus, we can then find ways to inhibit these behaviors in the pathogen, either by harnessing the potential of native coral-associated bacteria, or maybe by learning something more about coral immunity. If we can prevent the pathogens from establishing on the coral surfaces, that would be the first and the easiest way to prevent or stop the disease progression.
Experts are still in the early stages of research, but they hope their long-term findings will be a useful tool for future management and protection of coral reefs. In Key Largo, scientists from Florida International University are using underwater cages and garden fertilizer to gain a better understanding of the microbial health of corals.
It's kind of like a chessboard. When you go out there and dive on the reef, we've got essentially 8 different large plots. We have 4 with nutrients and 4 with no nutrients.
One of the major stressors on reefs is fertilizer that makes its way from land to sea. Dr. Darren Berkeypile and his team study how coral reef communities are affected by nutrients such as nitrogen and phosphorus.
Those are the 2 big key elements that humans impact the ocean by putting them in a lot, via fertilizer and sewage. So we essentially go buy common garden fertilizer that we put in PVC tubes. So we put these enrichment treatments at every corner of those 3-foot sections and then one in the middle.
Within these large plots are also experimental cages that keep herbivorous fish away from select corals.
They're essentially made out of plastic-coated chicken wire, and the mesh is about 1 inch in size, so the small fish can move through, but it keeps all the big grazers out.
By keeping grazers away and allowing seaweeds to grow at faster rates with the help of fertilizers, scientists are able to mimic stress factors such as pollution and overfishing. Using these experiments, Dr. Berkey-Pyle has observed a change in the mustard hill coral Porites astrioides.
Seaweed competition causes the Porites astrioides to lose an important bacteria. It's gamma proteobacteria. So this one bacteria is always found on Porites astrioides. It's thought that it's important for the health and growth of this coral. But when seaweeds start competing against the coral, this bacteria actually disappears from the microbial community.
Researchers are still investigating what the effects of this disappearance will have on the coral's health. Scientists from FIU not only monitor shifting microbial groups, but they also examine parrotfish for their potential to spread microbes.
So the parrotfish are important herbivores, but some of the parrotfish also eat coral. So we stretch barrier nets out on the bottom of the ocean, herd these parrotfish into the barrier nets with hand nets, catch them, take them up to the boat, and then we swab their mouths with a Q-tip, essentially, put it in a preservative, and then we can extract the DNA off that parrotfish mouth sample, see if we can match the bacteria on the parrotfish mouth to the bacteria on the corals that they just preyed on. They could take a bite of a diseased coral and potentially move a diseased microbe from a diseased coral to a healthy coral.
Determining whether or not parrotfish transport microbes from one coral head to another could give experts important insights into how coral diseases spread. Florida scientists aren't just studying the microbial communities of shallow water corals. Dr. Christina Kellogg from the U.S. Geological Survey in St. Petersburg, Florida, examines microbes from deep-sea corals.
In shallow water corals, if they're unhappy, it's like they change colors, they start to bleach. You can tell the deep-sea corals don't bleach, they're already white.
To access cold water corals from the depths of the Gulf of Mexico, scientists use manned submersibles. To collect specimens and prevent contamination, Dr. Kellogg created a sampling device which has been dubbed the Kellogg box.
It was a big acrylic box that could fit on the front of the submersible, and it had 10 separate bins. It's sort of like a little kid, you know, my spaghetti can't touch my sauce. Well, my corals can't touch another coral or sediment or water.
By isolating each sample, researchers can maintain the true microbial diversity of each coral and prevent cross-contamination of microbes. Once samples are brought to the lab, Dr. Kellogg can begin the process of DNA extraction.
And so when I'm ready to do extractions, I take the tubes out, keep them cold on ice, quickly measure out a very small amount of that powder, which that powder used to be the coral and the mucus and the bacteria. And you keep doing extractions where you make it purer and purer. And in the end, you end up with just pure DNA, and that's what we end up using to put on the microarray phylochips.
Initially developed by the Lawrence Berkeley National Laboratory to scan airborne microorganisms, scientists like Dr. Kellogg now use the phylochip to identify the types of bacteria found on coral DNA samples.
On those tiny little chips, which are like glass slides, robots spot little dots of DNA, which corresponds to about 50,000 different bacterial types. It allows me to get sort of a top-down view of the community that we haven't had in corals before. If any of the spots that are on the chip match a bacterium that's in my sample, then that spot lights up. The first way people looked at bacteria in general was to culture them. The problem is maybe 1 to 10% of the bacteria that are in the environment actually will agree to be cultured. And so you only see this tiny little subset of the whole world that's out there. The problem with disease is, what if what's causing the disease doesn't happen to be one of that 1 to 10%? You'd never know.
Experts believe it's important to know the microbes that live on corals and how they interact, because they are the earliest warning signs of serious changes on a reef.
If something changes in the environment, the water temperature or the level of nutrients, the microbial community will shift. By doing that, they're basically sort of our first diagnostic tool. Before you even see something with your eyes is wrong with the coral, you could see that change if you were looking at the microbiological level.
Major funding for this program was provided by the Batchelor Foundation, encouraging people to preserve and protect America's underwater resources, and by Divers Direct, inspiring the pursuit of tropical adventure scuba diving.